A very warm welcome, everyone. We'll be starting in more 15 minutes from now at the scheduled time. And thank you for joining. Meanwhile, we begin with today's webinar. It would be great to hear from all of you about your background. It could be institutional background or your research background, and also what are your research interests. You can put them in the chat box. Thank you. A very warm good evening, good morning, or good afternoon to everyone present here. Um, we'll be starting in more 10 minutes from now at the scheduled time. Thank you for joining. Good evening, Dr. Ajit. Good evening, Dr. Raghav.
Good evening, Sonalika. Good evening, everybody.
I think we're good to begin with today's webinar. So let me just start the recording. I would like to welcome you all for today's webinar on Introduction to Cheminformatics and Drug Discovery. It has been well established that discovering and developing drugs is an extensive and expensive process. However, since 2020, there has been a phenomenal demand to increase the use of bioinformatics, cheminformatics, and the use of machine learning and artificial intelligence for vaccine and drug development. With the rapidly growing amount of complex healthcare and biological data and the increasing complexities of analysis, everyday cutting edge innovations are bringing new things to learn. Hence, it is quite challenging for beginners to start their research journey in this fascinating field. And to overcome these challenges and bring high quality research training in the field of cheminformatics and drug discovery, we developed and designed an innovative and industry driven project based training opportunity for everyone. This program will be led by the mentors of Omics Logic Industry Expert Panel, which includes Pharma, Biotech, and Science Gurus, to help students, researchers, and scientists utilize structural biology and apply cheminformatics for drug discovery. This research training is designed to enable students to learn basic concepts of structural biology and to start applying by working and utilizing specialized algorithms and tools that have been utilized by industry and research labs to innovate, discover, and answer some of the important biological questions. So before we begin with today's webinar, I would like to introduce us. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. The training has been completed by over 24,000 participants from 187 countries in over 350 workshops. And due to this fast growth, our team is working with local and regional coordinators that are helping refine the local program logistics and leverage our online training resources, adapting them to the needs of students and researchers around the world. Over the past several years, we have had the privilege of working with a number of organizations developing a training program from research-oriented students, faculty, and democratizing bioinformatics, making this discipline accessible to thousands of students and faculty around the world. The research-based training programs are a result of close collaboration between Pine Biotech and numerous academic institutions that have participated in content evaluation, curriculum review, and project design. The training follows a project-based learning approach, which uses research-grade tools to analyze data from top peer-reviewed journals. It has been completed by students, researchers, and faculty in 6 different specialization tracks, which includes oncology, infectious diseases, precision medicine, neuroscience, data science for biomedical data, and comprehensive training on omics data analysis. I would like to take the honor to introduce the founders and team members of the company. Dr. Alfred Tauber comes from a medical background with an expertise in oncology, immunology, and biochemistry of information. And one of his major projects is the establishment of Tauber Bioinformatics Research Center, which has been at the core of what we do. Dr. Leonid Brodsky is the director of Tauber Bioinformatics Research Center and is an expert in bioinformatics and biostatistics. Our platform is a result of hard work by some of the people mentioned here on the right. We'll also hear by some of our mentors today in the webinar. And I am Sonalika Ray. I'm the Omics Logic Community Manager, and I'm responsible for daily interaction with thousands of our users, that are a part of a bioinformatics community, which is managed by Pine Biotech. Now, let's begin with today's webinar. So first of all, I would like to call on stage Dr. Puneet Kakkar. Dr. Puneet is an experienced, technology-oriented life science professional responsible for ideation of new capabilities and end-to-end management of existing scientific projects. He has an experience in serving clients coming from human drug discovery, veterinary medicines, and agrochemical settings. He's a strong advocate for using advanced technology in pharmaceutical research and development. We would like to hear from Dr. Puneet, mentor of the program. Over to you, sir.
Thank you very much, Sonalika. Just wanted to check if you can hear me loud and clear.
Yes, sir, we can hear you.
Okay, thank you very much. Thank you for all the participants joining this call. So in this Cheminformatics program, we will be learning and understanding a lot of traditional as well as new insights that are coming from the field of computational sciences as well as chemical sciences. In Cheminformatics, we will be talking about, initially we'll begin the journey with the chemical structures. So all the stories around chemical structure, which is the core asset of the pharmaceutical companies, and all the methods, including drug discovery overall process, modeling, simulations, applying machine learning approaches on the chemical structures, and thinking about something, what if we can automate the whole process of drug discovery? So in case the participants are coming from a varied disciplines, not having even a simple understanding of the whole drug discovery process or what the drug is, I believe this course will be very ideal for them. They will not only understand the drug discovery process, but they will also understand how to understand the basics data, which are driving force for the drug discovery data science practice, as well as applying tools and technology, understanding what is available free of cost, what is available on paid subscription base. All these tools, technology, database, their formats, understanding the data, and the overall process will be understood in this program. I will be taking a couple of sessions for the Cheminformatics and drug discovery, and I believe I should be the right mentor for those courses, because I come with a 15 years almost experience in drug discovery and development process, helping clients on multiple areas in Cheminformatics. Drug discovery and development is just one part where Cheminformatics can be leveraged. However, there's a variety of other disciplines where there is an increased demand of those who are skilled in Cheminformatics. For example, if you come from the IT background and you understand the system of cloud computing, cloud architecture, system architecture, and you are trying to help the clients in digital transformation of the in the chemical landscape at their end, certainly this course will be very, very seminal for you. This is extremely large field in the healthcare and life sciences practice, where digital transformation are changing the way scientists work into their day-to-day life. Doing hands-on on molecular modeling, simulations, running these screening campaigns for identification of hit molecules are just a couple of fancy, I would say very, very fancy topics that are always discussed in cheminformatics. But very fundamental is from where these chemicals usually come from. What is the origin of those chemical compounds? We will also understand how the data, what are the databases associated with these chemical compounds, what kind of informations are generally stored, when to rely on those information, how you can download those information, how can you consume those information. If the information is too large, what is the best practice to visualize those information? So all these informations are packaged together in a very nicely uh, uh, fashion, and this cheminformatics and drug discovery course is basically designed for that purpose. I hope you will find this course very interesting, and you will leverage the information for your in your current place. If you are a professional, you will be leveraging for your upskilling yourself into the better client deliverables. If you are a student, then you can upskill yourself for the job employability. If you are an entrepreneur, you can think about what are the solutions can be made and can build the startup. And if you are a PhD student or looking for a research areas or research topic, you will understand how to select, what to select, which topic will have a more demand in the coming 5 years, 10 years time, by the time when you will be graduated from the doctoral program. I hope in all the ways this program will be very beneficial to all of you. I will be mentor for some of the sessions and looking forward to meet all of you in the upcoming sessions starting tomorrow. Thank you.
Thank you so much, Dr. Puneet. And now I would like to call on stage Dr. Mohit Mazumdar to please introduce himself. Over to you, sir.
Thank you, Sonalika. Hello, everyone. It's a pleasure to meet you all, and I'm very happy to see Dr. Puneet Kakkar here with us, and Sonalika introducing all of us, and Dr. Ajit Kamath. It's a pleasure meeting you all, and I wanted to discuss let you all know a little bit about my background. So as a mentor, I've been working with Biotech for the last 3 years, helping students understand some of the important aspects of structural biology, and also applying some of these cheminformatics methods for their drug discovery projects. Along with that, we've been working together with Faba Academy, Science Gurus, and other academic partners, where we are developing resources and tools which will be really helpful for you to learn about cheminformatics and drug discovery that you learn throughout this program. So this entire program has been designed keeping in mind about different backgrounds of students coming from different educational systems, as well as keeping a basic understanding of what is going on in the industry. So as you have heard from Dr. Puneet, that you would be learning about some industry opportunities, some academic opportunities. So it is also important to understand that what are the different methods that industry is using for different problems. So in this program, we will be discussing that. And as my experience, I have over 30 publications in structural biology area, where I've worked with different academic researchers and scientists from all around the globe, focusing on finding solutions using computational modeling and molecular dynamics simulations, and finding specific key mutations to do to plan experiments, as well as to do some translational work to find some different types of variants and to look into their change in the active site. So in this program, we have also used all of those methods that is usually applied for cheminformatics, that is usually in application in research. So this will be a program which will be really beneficial for those who are coming to learn about cheminformatics as a application to drug discovery, but also structural biology and the different aspects of structural biology, and what this chemical data and protein information and biological information we can extract, and the mechanisms that we can look into using all these different aspects of structural biology and cheminformatics. So I'm very, very excited for this program, and I'm really looking forward to working with all of you with different ideas, working with all of you with different tools that we will be using throughout this program, along with this great mentors that we have along with us, which is Dr. Uday Saxena, co-founder and Regene's Bioscience, and he's a renowned professor, and he's been taking a lot of sessions, and we have the other mentors as well. So looking forward to meeting you all and then working through the sessions and the practical aspects. Thank you so much. Over to
Thank
you,
you so much.
Sonalika.
Thank you so much, Dr. Mohit. Now, I would like to invite Dr. Ajit Kamath, who will be talking about the advancements in drug discovery. Dr. Ajit has 25 plus years of preclinical drug discovery experience, primarily at Pfizer. He obtained his PhD in biochemistry from the Indian Institute of Science and carried out postdoctoral studies at the University of Texas, Austin, and Stanford University. Dr. Ajit joined Pfizer's R&D Center in Groton City and Cambridge and worked there for 13 plus years as an associate director. And after 20 years in the US, he returned to India and served as senior VP at Jubilant Biosys for 4 years. Dr. Ajit rejoined Pfizer as the head of external R&D innovation, worldwide R&D Pfizer India, and successfully initiated collaborations with Indian pharma companies and academia. His team contributed to 2 drugs, one in the market and one is in clinical trial. Dr. Ajit also worked at Mitra Biotech as executive director to lead their external research efforts. Currently, Dr. Ajit is an advisor to 2 startup companies, Pendorum Technologies, which focused on corneal blindness, lung and liver diseases, and Metastop Solutions working on cancer metastasis. He is the member of some of the DBT BioTech committees as well, which includes BIG, Bionest, and National Biopharma Mission. We are pleased to have you among us, Dr. Ajit. Over to you. Sparsh, I think you need to make Dr. Ajit co-host.
Sonalika, he's already co-host.
Thank you.
Can you hear me now?
Yes, sir, we can hear you.
Okay, yeah. So I'll start sharing my screen. Okay, can you see my screen?
Yes, sir, we can see your screen.
Okay, thank you. First of all, thank you very much, Sonalika, for the kind introduction, and Mohit, for all the interactions that Phaba Academy has with you and with Pine Biotech. Thank you. I would like to thank Pine Bio for this opportunity to interact with the youngsters. Potentially, they will all be joining your courses in Cheminformatics and take advantage of mentors like Mohit and Puneet and others who have agreed to put a nice program like this together. In my presentation, Mohit suggested that I keep it basic so that many of the beginners will be able to understand. So I will be talking about the recent advances in drug discovery and covering quite a number of aspects. So it's nice that all of you have an opportunity to learn during this course the power of Cheminformatics. It was not there, as Sonalika mentioned in the beginning. The technology has changed tremendously, and it's all for good. Okay, things have worked out very well. The technology is moving. Excuse me, moving so fast, and those things have helped drug discovery to go much faster than what it was, I would say, 10, 20 years ago when many of us started our drug discovery journey in companies such as Pfizer. So as Sonalika mentioned, until recently I was a senior director and head of external R&D for Pfizer. I'm also currently advisor for Pendorum Technologies and senior advisor for Metastop Solutions, which is working on cancer metastasis. Okay. The opinions and the content that I have in my slides, they're all my own, are adopted from the literature, adopted from publications, and they reflect my, my own ideas and opinions, and do not reflect that of my former or current employers. Okay. So what we'll do today is discuss about various aspects of drug discovery. Again, I'm trying to keep it basic. Certainly, we can get back to some of these, deep dive into some of these areas, depending on the interest at a later time. And we can certainly coordinate that with Mohit and Sonalika. So the topics I'm going to cover would be introduction to a drug discovery process. Let's talk about high throughput screening. We'll talk about structural biology and structure-based drug design, some of the selectivity screens that are employed in the pharma industry. We'll learn a little bit about biology, not much, and then talk about discovery of crizotinib, one of the kinase inhibitors for lung cancer, and then we'll summarize. Okay. But the message in all of these would be how fast the technology has changed and how fast people have been able to implement these technologies so that we'll be able to come up with medicines much faster for various diseases. Okay? And that is, I think, that is the theme of the entire cheminformatics course, where so much of knowledge, data that's being cranked out is analyzed very, very thoroughly, made sense of it, and then utilized for coming up with medicines. Okay. Drug discovery as such is a long process. Okay. It takes about 15 years, 10 to 15 years to come up with a new drug, and the entire process starts as shown here. Okay, you start with an idea. Okay, we can I can get into some of these interesting topics anytime in the future. So a team, what we call it, normally call it as a project team. I hope you can see the cursor arrow. Yeah. Okay. Yeah. Let me take a pen. Yeah, you can see the laser pointer, Sonalika.
Yes, sir.
Okay, okay. Yeah, so a project team, let's say a project team working on cancer meets together and say, this is the target that we would like to we would like to start with, use that particular target and develop a very, very selective and highly potent drug. So that's called the project team, involving lots and lots of scientists with the different backgrounds. Some are chemistry, some are biologists, some are pharmacologists. There will be, people specialized in ADME, safety, toxicology, all of them will be sitting together and saying, okay, how do we address this particular target and convert that into a nice, in this particular case, maybe small molecule, and take it into the patients later on. So if it is a small molecule, then, you go through synthesis, you go through a lot of screening. I think Puneet talked about it, and I'll come back to this at length during my presentation. And then, do a lot of discovery work. Okay. At the end of the discovery work, you start nominating a candidate after carrying out initial safety studies. And let's say you identify a candidate that is very, very safe, selective, and is ready to move into the clinical stage. Okay. So you carry out some more safety studies, long-term safety studies, and finally, you make it in kilogram quantities in a GMP manufacturing facility, and then try it in healthy patients. Sorry, not patients, subjects. Okay, these are volunteers, perhaps 20 to 30 subjects in this particular case who are participating in the study. In phase 1, you demonstrate the safety of that particular compound, and then you get into what we call it as phase 2, full development with 100 to 300 patients, and then if everything goes well there, then you move into another phase 3, which is the most critical part, ranging from somewhere between 300 to 3,000 to 10,000 to 30,000 patients. Okay, and these are real patients, and you analyze the data. If everything looks good, if it is very, very efficacious in phase 3, then you nominate that drug, and it is reviewed by US FDA, or in India, it's a CDSCO or DCGI, and finally, if all of them approve, the drug comes to the market. Okay, so this is the normal process of drug discovery that is followed. Okay. For a period of somewhere between 10 to 15 years. Okay, but the success rate in drug discovery is very, very low. Okay. If you start with 100 drug discovery programs, wherein your team of people a team of people working on drug discovery, and if you have 100 project teams working on 100 different programs, and you screen, let's say, 1 million compounds, when you talk a little bit about it, and then go through preclinical pharmacology, safety pharmacology, and then eventually you'll be able to get 1 or 2 drugs to the apologies, 1 or 2 drugs to the market over a period of 15 years. Okay, this is this is a challenge with drug discovery, a very, very long drawn process, and it takes around $2 billion for my colleagues, coming from India. This will be about 15,000 crores just to come up with one particular drug, taking 15 years. That's a very, very long drawn process with the about 2% to 3% of success rate. And many of us who worked in this industry for 20 plus years have essentially worked on about 100 different programs and then have 1 or 2 products in the market. So you can imagine the amount of time and effort that takes to bring one drug to the market. Okay, but this can't continue. Okay, things have to change, and this is where all the new technologies that are coming into the picture have helped to accelerate the entire drug discovery process. Okay, but why would the drugs fail? Okay, the drugs fail for a lot of reasons. The drugs are the diseases are much more complex. Okay, because we have a better understanding of the diseases. The drug is not supposed to stay too long in the body. If they stay too long in the body, again, it can cause toxicity. The drug failed. There will be some adverse reactions. There will be poor absorption of the drug, and there are instances of very low levels in the body. Everything looked good in animal studies, but when you get into the human body, the levels remain so low that that drug is no longer efficacious. So there are a lot of reasons why the drugs fail. And the most important thing that we'll discuss perhaps in my next talk on personalized medicine is the contribution from genetic makeup of the patient population. Okay, I think this particular factor most other companies did not take into account for years and years. We all thought that all of us are the same. If the drug works in patient A, it should work on patient B. If the drug works on people in the United States, it has to work on people in Europe, in India, in China, and everywhere else. But in reality, that is not true. And that's all because of the understanding we have about genetic variations among people. Genotypes are different for different people from different countries, different genotypic origins, and they all contribute to success of the drug or failure of the drug. And we'll discuss much more of this in the personalized talk that I'm going to give on Friday. So as I mentioned, in the last 15, 20 years, now, as we have the human genome sequence available, it has tremendously changed, in fact, revolutionized the drug discovery, because we know everything about a human genome. We know what are all its components, what are the genes that are there, what are the functions for most of the genes. Okay, how many enzymes are there in the human genome, what type of enzymes, how many receptors are there, the role of hormones. All these are very well understood now, thanks to human genome sequence available. And as Puneet mentioned, now it's a time of opportunity for all of us to incorporate some of the very new cutting-edge technologies into drug discovery. Okay, we have a better understanding about diseases. 10, 20, 30 years ago, when we talk about oncology, we talk about a very, very broad disease as oncology. But in reality, it is not. Okay, there are hundreds of types of cancers, and each of them have a unique way how that particular cancer started and how you can control that. It's not that cancer is one and then you have a simple drug that can, a common drug that will that can be used for all types of cancer. So we'll discuss more about this in my next talk. But the biology insights have been fantastic. We have very good targets. In the past, they were not druggable, but now they are druggable. Okay, so that also has contributed a lot. And then, of course, a lot of very powerful technologies such as cheminformatics, technologies such as robotics, technologies in high throughput screening. All these have really helped us to look at targets that were undruggable in the past, and now thanks to the technology, we are able to do much better in using these technologies to target those and then identify small molecule drugs. And now is the era of omics. Again, I think this workshop and the previous workshops arranged by Pine Bio will certainly help all of you to understand the role of each of these specific areas, whether it is genomics, transcriptomics, proteomics, metabolomics. All of these are very, very important in understanding how you decipher the disease and what type of molecules you can develop. Okay. Again, 10, 20 years ago, 30 years ago, these were all in nascent stage. People are not looking into these areas of omics, but now it is integral part of any of the drug discovery programs. So the course that Pine Bio is offering on omics is whether it is cheminformatics or the regular omics will cover many of these things, and you start appreciating as to how these technologies is going to help in drug discovery. In drug discovery, there are phases. We call it as the hit identification, target to hit identification, hit to lead, and then lead optimization, and then proof of concept studies, and eventually you nominate a candidate. At this stage, that candidate can get into the clinical study. Okay, so these are all the various phases. I will not get into the details of this, but once you start screening with the 100,000 compounds or 1 million compounds or 3 to 4 million compounds, at the end of it, you will end up with 1 or 2 compounds with a lot of confidence, and these molecules will get into the clinical study. That is phase 1 clinical study. Okay, so how does for scientists like us, how does this start at the beginning? I showed you one slide where the project team is sitting and discussing, how do we come up with the molecule, and this is what exactly they do. Okay, you identify an inhibitor which eventually will become the candidate for the clinical trial, and that is against one particular target, and let's consider that as a target protein. Okay, a target that has been implicated in one particular disease, and you want to inhibit that particular protein. So you produce that particular protein and then develop assays. You will see this in my future slides. Okay. Once you have that assays developed, you run a high throughput screen to come up with some molecules. Those molecules you discuss with medicinal chemistry. They'll make more molecules that are very selective and very potent. You use that knowledge to screen more enzymes, more targets, develop structure-based drug design effort, and then you go through selectivity screen so that your molecules are very, very selective. Okay, and then of course biology comes into the picture, in vitro biology, in vivo biology, that is pharmacology, and make sure that the compound is efficacious, the compound is very stable, not degraded, does not have any toxic properties. That is done using ADME and then PKPD studies and everything else. If it passes through all, then you have a potent preclinical inhibitor, preclinical molecule that you can take it for GLP tox studies and eventually take it into the clinic. And this normally takes 3 to 4 years, but thanks to all the technologies that that are currently available, it is about 2 to 3 years now, depending upon what type of target protein you are working on. Okay, so the same information in the previous slide, I've converted this into this slide, wherein we are talking about various components of drug discovery. There's high throughput screening, there's medicinal chemistry, there's structural biology and structure-based drug design, structure activity relationship, or we call it as SAR, Okay. Then of course, biology is there with the biochemical assays, cell-based assays, developing various in vivo models to show the efficacy of the molecule, pharmacology studies, PK/PD studies, and then of course, GLP tox study, which really tells you whether the molecule is safe or you have to kind of abandon the project. Okay. So in today's presentation, I'll focus quite a bit on high throughput screening, because the Cheminformatics coursework will cover some of these things as highlighted by Sonalika, Mohit, and Puneet. So we'll talk a little bit about it, and also I'm sure you will appreciate how things have changed for the better with regard to high throughput screening. We'll talk about structural biology and its impact on drug discovery. Again, you will learn a lot about this in the course, and then of course biology is important. You will hear some aspects of it in my presentation. Okay, I wanted to highlight the power of expression profiling in this particular presentation. Okay, so all of you are familiar with the DNA, that is genomics converted into RNA, that is transcriptomics. Then RNA becomes protein, and then is converted into protein, and protein has various functions, and eventually it has a role in developing all the metabolites that are used. So in the past, I think we were just looking at this alone, and then hoping that you will come up with the target protein and then inhibit that particular protein so that the metabolite is not formed. Okay, and that is essentially the small molecule drug discovery as an inhibitor. Whereas now, thanks to the technologies, all these components are utilized day to day in drug discovery process, whether it's genomics, transcriptomics, proteomics, and then metabolomics. Okay, it is no more saying that I have one particular target. You have to go back and see what does that particular protein do, what are its metabolites, what are its transcriptomics profile, how does the genome look, are there any mutations, what happens if there is mutations. All these things come into the picture, and that is what is demonstrated in this particular slide. Okay, so let's say we are trying to identify what is causing a disease. Okay, how do you understand the difference between a normal tissue or a normal subject versus a patient who has a disease? Okay, in this particular case, I'm giving an example of expression profiling done for breast cancer. Okay, in this particular case, you have the normal tissue. Okay, and this is the profile, expression profile of perhaps 20,000 genes. Okay, looking at all the profiles there. This is normal. And look at the cancer tissue, how overexpressed many of those genes are. Okay, and this is from the same patient. This is a neighboring tissue. Okay, this is the non-tumorous tissue, and this is the tumorous tissue of the breast, showing you, how many of these genes that are listed here are overexpressed, and they are the ones that are causing uncontrolled cell proliferation, leading to the tumor formation. Okay, and you can see exactly this what happens. Lot of pathways. I'm sure you will hear more about it during the course of this work, this workshop. Okay, various pathways involved in nucleotide biosynthesis. They're all overexpressed because the cells have to make so much of nucleotides for the growth of the tumor. Okay, so this tells you that there are opportunities for you in this particular case, inhibiting some of these pathways. Okay, using small molecules, and perhaps if they are working very, very effectively, then you'll be able to stop the growth of the tumor, and hence the patient can get better. Okay, so I just gave you that as an example to show how unlike what it was 10-20 years ago, how people are looking at targets, and now without data such as such as this one, it will be very hard to carry out any drug discovery work. Most of this, again, you will learn in this workshop, and it will greatly benefit you if you are taking up a job in an industry or in your research itself. Okay, let's quickly move into other aspects of drug discovery. HTS. Let's say we want to do a HTS for an enzyme target. We assume that this particular target is critical in one particular disease. Let's say cancer. It might be a kinase. There are a lot of enzymes. I'm putting this list because all of you may have worked on many of these or familiar with this. So, in the pharma industry, same targets are utilized for coming up with drugs. And in this particular case, let's talk about a kinase. Okay. So kinases have been implicated in various diseases, particularly in cancer, but they also have been implicated in neurology, metabolic diseases, inflammation, immunology. They're everywhere. Okay. But cancer is a place where, you know, there have been a large number of drugs for kinases have been developed, and they are currently, you know, being given to the patients as tyrosine kinase inhibitors, or TKIs. Okay, there are 518 kinases. Okay, and all of them have a role in various signaling pathways, which many of you have studied a lot, and also you will hear some of that in this particular workshop. So a protein is phosphorylated in the presence of ATP. You get phosphorylated protein, and this particular phosphorylated protein, you know, eventually phosphorylates others in the pathway, and eventually you will see the cell proliferation leading to tumor. That's an example that I'm going to highlight today. Okay, in this particular pathway, there are so many. I'm sure you have read about all these pathways there, MAP kinase pathway, and there you see so many targets have been identified. Okay, all these are kinase inhibitors, and if you inhibit this particular kinase, then, you know, the proliferation or differentiation comes to an end. That means the tumor doesn't grow, the tumor dies, so that, you know, the patient becomes normal. Okay, that's the hypothesis in which we work on, and these are all the various drugs that are currently available around the world, developed by various pharmaceutical companies to address various types of cancer. So we have about 80 different kinase drugs that are developed as of December 2021. Okay, I suggest you go through this review, Nature Review Drug Discovery by Phil Cohen, who is who is a fantastic leader in kinases. Okay, he wrote this review on on oncology drug development. Okay, so if you want to come up with a drug, let's get back into basics. Since most of you are beginners and students, I would like to emphasize how exactly the drug discovery works. Many of you would have cloned some genes, and that's what exactly happens in the pharma industry. You isolate a kinase gene that's of interest, and that particular kinase gene has a role in a given disease. Okay, you isolate it from cDNA, clone it into expression vector, either in E. coli, either for expression in E. coli or in baculovirus system or mammalian system. Express large quantities of cells, grow large amount of cells, and then purify the protein, develop the assay, you characterize it, and then in this particular case, use them for inhibition studies, but also for crystallization, which we'll talk a little bit later. Okay, this is to emphasize the purification. People use various tags, and I'm just showing you one example where we have purified large number of kinases. Okay, and I'm giving an example of some of the kinases that we purified: AKT, FA2, IRK, and CHEK2, and SULU, and STLK2, and various kinases. All of them have specific roles in cancer. Okay, so once you purify the protein, obviously, you would like to run SDSP gels, gel permeation to make sure that the protein is clean. Protein is pure. Okay, you run Western blots, you run mass spec to confirm the identity of the protein, N-terminal sequencing. You characterize the substrates, do all the enzyme characterization such as pH, metal, ATP, peptide chemistry, use peptide concentration optimization and other things. So there are multiple things that we'll have to do to characterize the target, the enzyme that are used for high throughput screening. Then you carry out the assay development. I'll not get into the details here, but this is, I think all of you have read about it in your coursework. So I'll skip this one, and I'll skip this one also. This is regarding IC50 determination. I just wanted to highlight that many of you have worked in the lab, carried out your research project, and this is not different. This is the same, except that in the pharma industry, you do it in a very, very robust way. In large number of molecules are being screened by the high throughput screening. Okay, high throughput screening, you not do it for million compound scale, and things have changed. People run 100,000 compound library, but others still go with million compounds library. You can screen structurally diverse sub-collections. You can go with the FDA-approved drugs or kinase inhibitors. You can go with natural products. And there are a lot of ways in which you can run a library. Okay, libraries against targeted kinases. You can run against proteases. You can run against phosphodiesterases. So there are subcategories of molecules that you can use for high throughput screening. That is with the assumption that more compounds you have, better chances you have of identifying a molecule. And that need not be true. Okay, you have to have a high quality compound that probably gives you high quality compound with a lot of diversity, has a better chance of success. And you have to have a good quality assays. Then again, you have high chances of success. Okay, and then you have library. We call it a singletons and compressed library. Some of it, I think, may be covered in the workshop, Cheminformatics workshop. Okay, so if you are running high throughput screening, then you have the assay developed. We talked in previous slides how we develop the assay. Now you have the library. You screen against these millions of compounds in the primary screening. Okay, and then you recheck it again. We call it as retest or confirmation. Concentration dependence, that's IC50 determination. And finally, you come up with the molecule. It's a lead molecule, but at this stage, we call it as a hit. And then you have to improve the property before it becomes a lead. And eventually, you'll be able to carry out the efficacy study on these molecules. Okay. How do you run high throughput screening? Okay, and how do you analyze the data? And that's where all these technologies come to the picture. There are a lot of spectroscopic techniques such as UV, fluorescence, bioluminescence, chemiluminescence, and technologies that you can use for running screen. People use mass spec, people use GC, HPLC. Various technologies are available in addition to the very well-known radioactive assays. People are going away from radioactive assays because of potential effects of radioactive materials. It's all people focus more on non-radioactive methodologies, particularly spectroscopy, if you can adopt that. Okay. And now comes the data. Okay, Puneet talked about it. I'm sure you will hear much more in the workshop. How do you analyze this data? Okay, let's say you have 1 million compounds. You have so much data. 1 million compounds, perhaps from say 1,000 or 2,000, 3,000 chemical series. Okay. In fact, it will more than that. Okay, so you have thousands and thousands of molecules. Okay, you want to visualize all of it. This is actually, for most of us biologists, this is something we always wait for. Okay, when the chemists are, when we run all the screen, the chemists are sitting together. The Cheminformatics, everybody's sitting together and saying, okay, tell me. Okay, you ran against 1 million compounds. Tell me the best molecules. How do they look like? Okay, show me the best inhibitors that are there, best series, which we can work on. So this is answering, months and months of work of all running the screen and everything else. And this is essentially the type of output you would like to that you get. And then then the Cheminformatics team and rest of them sit together and say, okay, from this, the following molecules, following series are good, and you can adopt that and then put more effort. I just wanted to highlight this particular heat map so that to tell you how things have improved tremendously in the past few years, wherein we are visualizing million compounds in one particular slide. Okay. The limitations of high throughput screen requires very, very expensive instruments. The reagents are very, very expensive. And of course, if you're running million compounds, I mean, a lot of time is also required, though, things are being miniaturized now, and you'll be able to run one particular screen perhaps in 1 week with 1 million to 2 million compounds. But still, it's a lot of work. Okay. This has been going on for years, and recently people have moved on to develop libraries using DNA-coded libraries, how they have revolutionized the drug discovery. Okay, I want you to go through some of these fantastic technologies that are coming up, wherein you are able to screen, 40 trillion molecules. All these are DNA-encoded libraries. And they're developed by companies such as New Evolution, and these are being used now for drug discovery. Okay, and I also wanted to quote the CEO of the company saying that don't think 40 trillion is a large number. You can put all of them in a front of you. Okay, so and then you will be able to screen. And the recent studies do show that there have been successes of such an approach of DNA-encoded library. And these are the 2-3 molecules from GSK that are in the clinic, and from AstraZeneca that are in the clinic, all started using this particular technology. You can do a Google search for this particular technology if you're interested, and you will get to know more as to why DNA-encoded libraries have become very, very popular. Let's look at one more example of doing the previous one is a real library. Okay, and this particular case is a virtual screening with the 3 trillion compound library. It's a cutting-edge technology developed by a company in California called Atomwise. Okay, in collaboration with a company in China called Senho Pharma. Okay, in this particular case, they do a lot of in silico screening. Again, in this workshop, you will get to know a lot more from Mohit and others about the power of in silico screening. In this particular case, they don't need crystal structure, which is very good, because most of them have crystal structure, but without that also you'll be able to use it. And you don't need local ligand training data. Some of these are proprietary technologies that they're developed, but supposed to be giving them fantastic results. And all of it is done by screening these trillion compounds in silico and come up with a molecule. Okay, and these molecules are synthesized in the lab. Okay, so from 3 trillion compounds virtual screening, you come up with perhaps about 1,000 compounds, which are very good. You go back to the lab, work with the collaborator, and they'll make you those 1,000 compounds. You test those in the lab, in the assay. Okay, again, all these technologies are possible only using tools that I think you will hear more about it. You will learn more about it in the Cheminformatics workshop. Okay, so we looked at one aspect. I may need about 15-20 minutes. I hope that's okay, Sonalika. Yeah. I'll cover some aspects of, in addition to structural biology, which is one approach. The other approach is looking at structural biology and structure-based drug design. Okay. If you have the structure of a protein and you know where exactly the compound is binding, then you will be able to develop molecules rationally. You can design molecules that bind very tightly and very effectively. In this particular case, this is the binding site for a kinase, and you will be able to develop molecules. And this is the approach. You will hear more about it in the workshop as to how this also has greatly helped in developing molecules for various diseases. I'm giving an example of kinase A. In this particular case, this kinase A was expressed in E. coli, purified, crystallized. You solve the structure, and here is the structure of this particular enzyme. That is, it's a kinase. It has a binding site, and this is where this ATP binding site and the inhibitors bind in this particular site, and you will be able to look at the active site residues and then design the molecule that will bind tightly here. Okay, and then you can make a lot of analogs around it, and then try to come up with the molecule that will essentially that will be a very potent selective molecule. Okay, another example for you, another kinase, again purified, crystallized, and structure is solved by X-ray diffraction. Okay, and then you get to see where the inhibitor is binding. Okay, few more examples of protein purification and getting crystals and solving structure. Okay. All this is possible because of the recent advances in technology. When many of us started working on this 10, 20, 30 years ago, we would clone 2 genes, 3 genes at a time. But now you have robots that can do high throughput cloning, robots that can do high throughput cell culture. You don't have to grow one clone at a time. You can do 96 at a time. Okay, if not more, you can purify 96 proteins at a time using various technologies. And then you can use crystallization machines that can crystallize nanoliter samples and then try to get to the crystal structure. And then you can use synchrotron. This is an example of synchrotron in UK, where you can ship your crystals and they will collect the data for you. And in fact, you can sit in your office here in India or wherever your lab is, and you can collect data remotely. Okay, so that is the power of technology that is helping us to come up with drugs much faster. Okay, so you identify, in this particular case, I'm giving an example of a kinase. ATP binding site is binding here, and these are all the interactions that are there for this particular inhibitor with various residues in the active site. I'm sure you will hear much more about it in Mohit's presentation. Okay, this is a PLK1, a project that we worked on many years ago at Pfizer. It has an N-terminal lobe and then a C-terminal lobe and then activation lobe. Okay, and this is the ATP binding site. Okay, I would like to get into some details here. This is the ATP binding site of PLK, one of the enzymes that is targeted for cancer. But you would like to identify molecules at a selective site, selectivity site, or activation loop site, or DFG in, DFG out, so that you will get more selectivity. This is classical site. This is non-classical site. Okay, because there are so many other kinases that have the ATP binding pocket. Okay, getting selective molecules are quite challenging for kinases, but people have been able to get it. Okay, there are 518 kinases, and all of them have the same kinase ATP binding domain. So the residues will be similar. So how do you get selectivity? So that's a challenge. There are 2,000 ATP binding proteins. They have the ATP binding pocket too, similar to kinases. How do you get selectivity there? And then, of course, there are multiple phosphorylated forms, non-phosphorylated forms, somatic mutations, alternate pathways. So though we think that we know kinases very well, getting a selective inhibitor for kinase is always a challenge. Once you have an inhibitor towards a kinase, you always develop something known as kinase selectivity screening. Okay, so that you will be able to make sure that the kinase that you are inhibiting is selective only for that particular kinase, and it does not hit remaining 517 different kinases. Okay, I'm giving an example of using 50 kinases, okay, for developing a selectivity screen. These are all the examples. This is a kinome tree, which I'm sure you are all familiar. you pick up, you identify some of these kinases that are important for your disease, but also you want to have some kinases you don't want to touch, kinases that have a role in kidney function. You don't want to inhibit kidneys function. You don't want to inhibit cardiac function. Many kinases have a role there. So you want to make sure that your compound is selective to one particular kinase which you want to inhibit, but does not inhibit other kinases. Okay. So imagine you have 1,000 compounds or 10,000 compounds you are running against. In this particular case, I've given 50 kinase examples. Assume you are running against 300 kinases. Look at the data that you can generate. Okay, again, cheminformatics come to the picture to identify, how would you go about identifying which inhibitor binds to which of the kinases and causing the least side effects. Okay, this is just one of the heat maps that you can see, but imagine running this heat map with 1,000 compounds against 300 kinases. Okay, and people have done that. Okay, so that is the power of cheminformatics. That is the power of knowledge that's available for accessing drug, for accelerating drug discovery. Okay, imagine having all these components in your hands. You have a targeted library for kinases. You have we talked about kinase selectivity panel. You have focus structure. You know where exactly it binds, how it binds, and why it binds. More importantly, if you know all those things, and then of course, if you know all the mutations that are there, all of it becomes very, very critical for coming up with the molecule that is very, very potent and very, very selective. And then only that particular molecule is nominated as a drug. Okay. We quickly go over the animal models without getting into the details. Once a molecule is identified as very, very potent and selective, you go into testing their efficacy in animal models. You carry out in vitro studies, some of the in vitro studies with the cytochrome P450s and other things, and then in vivo studies as shown here. This is a mouse model that is commonly used. Okay, you you inject a tumor and create a tumor in that mouse. Okay, and now you inject this particular mouse with the drug that you think is very, very effective against this particular cancer type. Okay, and if the drug is very effective, then the drug, the tumor size has to come down. The tumor has to essentially disappear. So you carry out lots of histopathology studies. You carry out ELISA, histopathology studies, ELISA, and everything else to see whether the drug is effective or not. And if the drug is really good, this is the type of data you would like to see. This is untreated animals. Okay, the mouse model. Okay, you can see just the vehicle alone. Okay, there's a control. That the cancer is there everywhere. Whereas you treat it with the drug such as Sutent. This is a Pfizer drug. Has been there for the last 15 years or more. Very, very potent, effective drug. You can see that within a short period of time that the tumor just disappears. Okay, this is the type of molecule we all aspire to develop in the pharma industry. Okay, I'll quickly go through a few more slides. Okay, this is development of a molecule and discovery of c-MET inhibitor, a drug called crizotinib. Again, developed at Pfizer for lung cancer. Okay, I want to show this particular case for you because you're interested in taking the cheminformatics course, and it all is related to this particular drug discovery is related to structure-based drug design, wherein the scientists had developed c-MET structure. Okay, they had access to three-dimensional structure of c-MET. They knew where exactly the compound binds, and they used this particular information to make about 800 compounds in less than 2 years. Okay, this is amazing. Normally it takes 4 to 5 years to have a program like this, and you will hear more about this in my presentation on Friday in personalized medicine. Okay, so using structure-based drug design, they were able to come up with the molecule. This original molecule, you modify, and finally come up with a molecule that eventually became the drug. Okay, the drug was very, very effective. It is available everywhere in the world. Okay, you can see the difference. This is untreated, and this is treated. This is untreated lung cancer. This is treated. You can see, and then of course, it's also effective. This is untreated, and then after treatment, the tumor regression you can see, saying that that the drug is very, very selective and very effective against lung cancer. Okay, but then you start developing mutations for any given tumor, and you have to develop next generation drugs. Okay, we call it as first generation drug, second generation drug, and third generation drug. Okay, the example I give you, crizotinib is a first generation drug. It works with some mutations, but not all the mutations. Okay, but then the scientists from Pfizer and Merck and GSK, they all came up with molecules that are better than the first generation molecules, the second generation compound, and then of course we have lorlatinib again from Pfizer, which is probably the best third generation drug that's available for lung cancer. Okay, the advantage of this is shown in this particular cartoon here, okay, that I'm showing with the pointer. Okay, in lung cancer, when it reaches at a late stage, it can metastasize. It can go to the other organs, and in this particular case, it goes to the brain. So you start seeing metastasis in the brain. This particular drug goes to the brain. Okay, unlike many drugs that does not enter the brain, lorlatinib goes to the brain, so it is effective even if there are some activities for this particular drug in the brain. Okay, which is very good. Okay, so that was the last slide I had, wherein I told you the story of how we develop molecules, starting from idea, okay, going through cloning, purification of the protein, developing the assays, and then running high throughput screening. In parallel, you get the three-dimensional structure of the protein, you do structure-based drug design, and then develop a lot of molecules, in a rational manner, and then take it through animal models, demonstrate the efficacy of those molecules, demonstrate they're not toxic, they're very selective, and then eventually, if they're not toxic, if they're very safe, in the GLP tox study, then you nominate them as a candidate ready for clinical trials. It goes through clinical trials, and if it succeeds, after 10 years of hard work, 15 years of hard work, it comes to the market. Okay, and then it benefits the patient. Okay, so that is in brief the entire process of drug discovery, and I'll stop it here and then perhaps take some questions, and then we can get into the career opportunities in drug discovery area or in cheminformatics. Back to you, Sonalika.
Thank you so much, sir, for such a wonderful session. And we are definitely looking forward to the session on April 22nd, where we are going to get some more insights about just the topics that you discussed with us today. And do we have any questions? Please put them in the chat box. Okay, so if there are no questions, I think we are good to go ahead. And now I would like to invite Dr. Raghavendran. Dr. Raghavendran will be helping us to understand the structure of the whole program, and he will take us through the curriculum. Over to you, sir.
Thank you, Sonalika. I think Dr. Kamath is still yet to discuss career opportunities, which he said he will after attending to some of the questions. Is that so, sir?
Yeah, we can take some questions, and I need maybe 2 minutes to highlight the importance of this particular workshop and the opportunities, in the pharma industry.
Yeah, please go ahead, sir. Please go ahead, sir. We can take the questions as they come up.
Okay. Yeah, see, for me, the cheminformatics and the type of tools that are available is really, really a blessing for people in drug discovery. Okay, it has, it has so many components that are useful in the pharma industry or biotech industry or in academia or wherever. Okay, that you are working or wherever you are trying to make your career, either pursuing your studies further or getting into industry and then working. Okay, and these are the things that everybody in the industry will be looking for you to know, how you can apply many of the tools that Mohit, Sonalika, and Puneet, they all talked about. They are very, very important. And I think through these workshops, you will learn about data analysis, okay, which is part and parcel of, I gave high throughput as an example, high throughput screening as an example, but data analysis, the type of data that are used in the industry with 100 compounds against 200 targets, 1,000 compounds against 100 targets. All these are done by using technologies that are being taught to you in the Cheminformatics coursework that we're talking about. In silico screening, again, it's part and parcel of any of the pharma industry, biotech industry, and you will learn a lot about this during the workshop. Structure-based drug design, structural biology work, homology modeling, okay, the docking aspects of it. Again, you will hear a lot about this. You will learn a lot. Perhaps you may get some hands-on experience of doing this during this workshop. All these things are very, very critical. And don't assume that it's required. All these things are useful only in the pharma industry. Okay, I think Puneet talked about it. These are the tools. Okay, in the industry or wherever you're trying to look for a job or in your own career in academia, these will be useful tools. Okay, doesn't matter how you want to utilize it. The fact that you have knowledge about all these things, then it's up to you how you want to use it in your own area, whether it's in life science discovery or whether it's in agriculture, veterinary, or in your own research, where you will be able to use these tools to understand the role of hundreds and hundreds of data points. Okay, so I think for me it's a very, very important workshop that you are looking into participating, and it will greatly help you. It will bring you up to speed as to what's going on. You have access to all the tools and everything else that others have. Okay, so my suggestion is take advantage of it. You'll learn a lot, and it will improve your profile. So I'll stop it there, Sonalika and Raghavendra. Thank you.
Thank you very much, sir. There are a couple of questions if you have a couple of minutes more to address them. There's a very broad question from Sailaja, who asks some of the reasons for disapproval of a drug from FDA.
From
FDA.
Oh, yeah. No. This is a very good question. Everybody looks for safety. Okay, the molecule has to be very, very safe. Okay, people are taking medicines for 20 years, 30 years. If I have arthritis and I'm living for 20 years, 30 years, the drug has to be safe for that long. Okay, so it is very, very critical for the FDA to make sure that the drug is very, very effective, is safe. Okay, effectiveness comes later. Okay, if the drug is not effective, then nobody will buy that drug anyway. That's for the pharma to make sure that it is effective. But making sure that the drug is safe is the reason why FDA takes a long time to approve it, and the rules are very, very stringent.
Thank you very much, sir. That perfectly answers it. And one more carrier question from Manpreet Kaur. She says she's a small molecule crystallographer with a PhD and a master's in data science, and she wants to get into data science-based roles in the drug discovery process. So what are your suggestions or advices for her to focus on?
No, I think data science is coming up, and there are so many companies in India working on it. I don't know whether you are looking for an opportunity in India or outside, Don't narrow down yourself, or for companies that are working on structural biology, the data science component itself would be of lot of interest to companies. In India, companies such as I can talk some of them which are doing a lot of crystallography studies, but a lot of screening work, a lot of bioinformatics and cheminformatics companies such as Syngene and Accelera, and there are so many other companies that are doing it. Origin and GVK and Jubilant, and there are quite a number of companies. Syngene, again, working on many of these aspects. So I think opportunities are there. You'll have to just apply to so many of these places, and then have a good interview.
Thank you, sir. I think, Manpreet, that answers your question. You have all the ingredients that is necessary. Please go ahead and look at the necessary all the companies listed by Dr. Kamath, and you should get a lot of success. I think that is going to be all with the question. And if it is possible, can you share your contact? There is a specific question to you from Samia, who wants to work with you. Maybe she can get in touch with you in person.
You can give my contact information to her.
Sure, sir. Sure. Thank you very much, Samia. I will share it with all of you in a while when Sonalika takes the stage. Thank you very much, Dr. Kamath. That was extremely insightful and amazing talk. It's a pleasure to listen to you.
Sure. No problem. I think I'm happy that you are arranging this workshop. For a person like me, none of us had this access 10-15 years ago, and now this is, everybody's on par in India or anywhere else, developing world. We have the same tools, same access, same knowledge as what's in the West. So I think it should greatly help all the participants to participate in your workshop and contribute to their success.
Thank you very much, sir. And if I may, if you may stop your screen sharing, I can start with mine.
Sure. Okay. Yeah.
Thank you, sir. So with that wonderful lecture and wonderful talk and extremely informative talk from Dr. Kamath, I want to take this opportunity to thank Dr. Kamath one more time. And I would like to introduce myself. I'm Raghavendran, bioinformatics mentor with Pine Biotech, and I will take this opportunity to welcome you all for today's session one more time. We are going to introduce you to this program, Introduction to Cheminformatics and Drug Discovery. And we already had an exciting and informative talk on the advancements of drug discovery by Dr. Kamath, and you already had met Dr. Puneet Kakkar, who will be one of your mentors for this program. And to begin with, let's go over the program in general. So before even starting to discuss about the program, I want to point a few things about how scientists and researchers are harnessing big omics data and advanced tools like artificial intelligence for drug discovery. So the past few years, we have witnessed the generation of big omics data across multiple modalities that include phenomics, genomics, transcriptomics, epigenomics, and metagenomics, to name a few. These omics fields are based on high throughput technologies like next generation sequencing technologies. And these technologies generate billions and billions of data points on genes, proteins, and other molecules that characterize the biological systems and biological phenomena. So this big data brings new hope and also new challenges in translating these data points to therapeutics. So we all know multiple targeted therapies in the past have shown efficacies without a well understood clinical biomarkers specifically for response or resistance. So what these multiomics provides is this systems approach, multiomics based systems approach aims to target a list of diseases or list of molecular features such as transcriptomics or gene expression signatures, as expertly described by Dr. Kamath. This can be used to complement the conventional target-based approaches. So we also know that emerging artificial intelligence methods that include deep learning and machine learning could empower the use of big data in both the therapeutic discoveries and to identify predictive biomarkers for patient's response or resistance to a specific target or to specific treatment. So how does these omics data in general empower us in drug discovery? So there are various types of omics data that describe in great detail the intracellular and intercellular processes associated with common diseases. So these data sets can be analyzed to understand the biology behind the host factors. And these data sets can even describe the microbiome composition of our bodies. And these data sets can also describe the environments we live in to drive infection and cause deterioration of health, to destabilize the immune system, and to lead into clinical manifestation of what we call as diseases. So developing a thorough understanding at the systems level of the disease is going to complement, and in fact, it is going to, it has already started to drive the conventional and the well-established methods of drug discovery processes like target-based approaches. So with that context, in that context, this particular program is actually designed for beginners and students who are eager to learn about the fascinating field of cheminformatics, drug discovery, and how computational structural biology plays a major part in these research fields. The aim of this program is to enable students and researchers to use open source tools, to use or utilize, to leverage public repositories, to gain more insights, and to develop quality research projects to answer key questions in structural biology, for example. So interested students and researchers will also have opportunity to learn about the application of these logic in programming tools and environment like R and Python for implementation of AlphaFold, for example, to predict protein structure. So what does this program cover, and who will benefit from such a program? So we start from absolute basics, like basics of structural biology. What are molecules that make the cells? What are they made of, and how they interact and how they function? So what techniques do we use to understand how thousands of different molecules in each of our cell work together to keep us healthy or to define a phenotype of the cell? And in addition to this, you will also learn about different kinds of protein, which is a macromolecule that aid biochemical reactions or specific function that will help the cells to hold their shape, or some kinds of proteins also help transporting specific molecules in and around the cell, etc. You will also learn how the structures of these proteins are determined, how understanding the relationship between the structure and function will enable us in designing drugs based on specific structure of the target protein. So in this context, you will also get introduced to essential methods like structure-based drug design, and how it is becoming an essential tool for faster and more cost-efficient methods to discover new lead molecules. So protein structural studies have provided hundreds of new targets and opportunities for future drug discoveries. I mean, hundreds is a very conservative estimate. The speed at which these structural investigations have occurred has been breathtaking, as we all know. Simultaneously, the availability of these data from Protein Data Bank, repositories like Protein Data Bank and other sources has benefited us in the long history of making structural data and metadata accessible to the world wide community. So you will learn how to analyze these publicly available structural data to reveal interesting insights into the three-dimensional structure of protein targets that are extremely critical and important for the discovery of new drugs. So we all know about the Protein Data Bank. To tell a little bit about the Protein Data Bank, a small statistics will reveal that the Protein Data Bank archive currently holds more than 155,000 structures at the atomic level of biomolecules. And these structures have facilitated the discovery of about 90% of the new drugs approved by the US FDA in the cycle of 2010 to 2016 alone. So that will actually tell you or underline the importance of using structure-based drug discovery in the context. So this program, as you have been listening to it for a lot of times, will be led by the members of Omics Logic Industry Expert Panel like Fab Academy, Pine Biotech, and renowned scientists from Accenture and Reagene Biosciences. So this research training is designed to enable you students to learn basic concepts of structural biology and to start applying by working and utilizing specialized algorithms and tools that have been utilized by industry and research labs to innovate, discover, and answer some of the key biological questions in cheminformatics and drug discovery. So, so this program has already started with an interesting talk from Dr. Kamath, and that would have actually given you a lot of information to process. So let's go over what is in schedule for the future topics and future sessions in this specific program. So this will be followed by tomorrow's session on Introduction to Cheminformatics by Dr. Puneet, who you met already earlier. So you will learn an introduction to structure, interaction, and properties defining the structure and interaction of molecules in general. You will also get insights into the drug discovery pipeline and how to use various tools in the progress or in the process of this pipeline. So this introduction lecture will be followed by an invited session that is open to all. This industry-based or industry expert panel session is scheduled for 2 days starting 21st of April and 22nd of April. So the first target is on drug repurposing by Dr. Uday Saxena on drug, on the topic of drug repurposing, which is also sometimes referred to as drug repositioning. So this is a process of identifying novel therapeutic potentials for already existing drugs and discovering new therapies for untreated diseases. So since this specific drug repurposing work or studies relies on data for existing drugs and diseases and data that and data that will fuel enormous, or that is fueled by the enormous growth of publicly available large-scale omics data, biological data, biomedical data, and also electronic health-related data. So the availability of these data, the availability of the methods that are associated with advanced statistical methods like artificial intelligence and machine learning, with the access to high-performance computing, has actually fueled or accelerated the development of these computational drug repurposing approaches. So this session will be an excellent start for you if you are specifically from, or if you are highly trained in data science and you are looking forward to use a lot of machine learning and AI-based tools to drug discovery and other purposes. So this will be followed by the next day's talk on precision medicine by none other than Dr. Kamath, who you have listened earlier, and he had given some key highlights or some key clues to what he will be talking about on the 22nd of April. So we know that what precision medicine is, as precision medicine marks a new relationship between genomics and drug discovery. This relationship will provide insights both into the mechanism and potential treatment options of a single patient's disease, and with that precision. So you will listen and learn a lot more on these two industry expert sessions, which is scheduled for 21 and 22. Please attend without fail. And then we follow this up with our scheduled third session by Dr. Puneet Thakur, who is going to be your primary mentor for this program, progressing from that day. So this is scheduled as a very insightful session on planned as an insightful session or a hands-on session on molecular modeling using Swiss model and AlphaFold tools for three-dimensional structure analysis of a binding site analysis and prediction of structures for the new sequences or other proteins. So you will also get introduced to tools and databases like Modeler, Swiss model, AlphaFold, Chimera, SwissProt, and EMBL repositories, to name a few. And then we will have a next session which will help you to find, download, and explore relevant data that is extremely critical to do target-based drug discovery. What will you learn in this session? You will learn about big data in terms of navigation in chemical space. You will learn about molecular representation in AI-driven drug discovery, and you will get an overview of application of these artificial intelligence and structure based drug design and virtual screening of these hits that you have designed and done. So the next session in this program is now scheduled as a practical session or a hands-on session where you will learn about introduction to structure, chemical interaction through docking. And you will learn also how to use public information of small molecule structures and properties to screen ligands based on its effectiveness in binding or based on its binding interaction or interaction with specific target that you have already selected. And the next session is actually planned as an advanced session in advanced cheminformatics. So where you will specifically learn how to use open source tools like data warrior to calculate and to predict key properties like drug likeness or LLE to identify new leads and novel chemical representatives of the leads that you have already shortlisted. So the whole program is then planned to culminate into an industrial perspective of how to design a cheminformatics research project and to address the opportunity to enroll yourself into a full drug discovery omics logic research fellowship program on 7th of May. And to top it off, to top this program, we have a final session which is planned as a panel discussion on the scope of cheminformatics, on the scope of bioinformatics and drug discovery in general. So this panel, which will include renowned scientists and mentors that you have already listened to, will discuss opportunities present in the industries and what are the skills and what profiles are in high demand. And this panel is also will have specific time scheduled to be an interactive session with an aim of addressing current opportunities and career options for students who are interested in the field of drug discovery in general. So with that, I take this opportunity to once again welcome you all to sign up for this highly informative program, and I look forward to meet you all in the upcoming sessions. Thank you very, very much for your participation, and I hand over the podium to Sonali. Thank you, Sonali.
Thank you so much, Dr. Raghav, for explaining the whole curriculum. Now I would like to share my screen. And Manpreet, to answer your question, yes, you can definitely still join the program no matter if you are in a different country. So we'll be able to then send you the recordings of each and every session, and you can go through the recordings, and we'll have a group. I'll just explain you all about how do we stay connected, how can you all take questions from the mentors. So we have a Slack group, a Skype group, and you can join these groups, go through the recordings at your own pace, and then come up with your questions. And the mentors will be able to guide you with that. All right. So and now it's time that I explain the registration process to all of you present here. I know many of you are really excited to get started with Cheminformatics. So I'll quickly put up a form, first of all, in the chat box. Those who have not filled the form, if you're interested to register for the program, please fill this form. The second thing is, here is the link to the program page. Now on this program page, you'll be able to, first of all, look at the whole schedule of the program that Dr. Raghav just explained. So the whole schedule is mentioned here on the program page. You can go through each and every session detail here. And then to register yourself, scroll down on the program page itself, and you will be able to find 3 different levels: beginner, intermediate, and advanced. So you can enroll picking up any of these 3 levels that works the best for you. Now, what are these 3 subscription levels all about? In the beginner level, you will have the access to such sessions, these group sessions. You will have the access to where you can listen to the mentors recordings, and also the same access will be given to each one of the person in these 3 different levels. In the intermediate level, you'll get a chance to have one-on-one meeting with the mentor to maybe clarify your questions or to clarify any of your research question, even if you're working on it. And then comes the advanced subscription, where you'll get some extra time apart from the whole duration of the program itself. And in this extra time, you can work on a nice, good research proposal with your mentor. So these are the three differences in these three subscription levels. You can pick the option that works the best for you and enroll yourself. So that was it about how you can register for the program. Now, it's broken for you. Just a second, Manpreet. I just send the link once again. And also, if you are not able to fill the form, maybe you can reach out to us directly at support@pine.bio or marketing@omixlogic.com. All right. And the question that Manpreet asked about her being in US and not being able to attend the sessions live, the same thing applies to others as well, those who are in different parts of the world. And if the time doesn't work for you, let's say, then you need not worry because we have the recordings definitely. We can do one thing for such people. You can go through the recordings and maybe we can have a live Q&A session at your scheduled time. Like you can propose a time to us and then we can finalize a particular time where we can meet and we can take your questions that you will come across by going through all of these sessions. All right. And by talking about the recordings, you will be able to also look at these recordings here on the program page itself. So what I'll be doing is I'll be just putting up the recording of each and every session here on the program page, and you can revisit the recording, go through it, and then click on the complete session. All right, so that was about how you can register for this Cheminformatics and Drug Discovery program. And tomorrow we have a session by Dr. Puneet. So this was a free webinar. Also, I had a question in the chat box asking if this is a free webinar or is it included in the program. It's a free webinar where we just wanted to introduce the whole structure of the program and also wanted our mentors to introduce themselves. All right, so now I'll just quickly tell you more about the Omixlogic Learn portal and also help you navigate through this portal. And along with the program that we have, Introduction to Cheminformatics program, we'll be having various associated resources that you all will be supposed to go through. And I'll just take you through this particular coursework as well, that is on structural biology and cheminformatics for drug discovery that you are supposed to go through along with this particular program as an associated online resource. All right, so I'll just share the link in the chat box of the Omixlogic Learn portal. Okay, Raghav sir, I'll just send the link of the form once again in the chat box. All right, so this is the link to the Omixlogic Learn portal in the chat box. Once you visit this portal, you need to sign up and create an account for yourself. It's very easy. You just need to create an account, putting up the credentials, or you can simply sign up by using any one of your social media accounts. It could be via Google, Facebook, LinkedIn, etc. So I'll just quickly give you a demo. You can create an account or use any of your social media accounts. I'll use my Google account. You can also follow along and have an account for yourself on the portal. Once you have an account on the portal, please make sure that you update your profile. Add a profile picture, your name should be in title case, add a brief bio, and also you can link in your social media accounts. Now, I'll be taking you through some of the courseworks that you can go through by having an account on the portal and also by being a part of the program. Now, each and every coursework that you complete, first of all, you can monitor your progress here on your profile. And for every coursework that you complete, you'll get some points that will take you from a beginner's level to an expert level, right? And for every coursework that you complete, you will be able to get a certificate here itself in your profile tab, download it, and share your achievement on social media platforms. All right, now let's go back to the courses tab. Now, how do you navigate the portal? So we have various courses and also example projects here on the portal that you can go through and learn more about. So if you click on the courses tab, you'll find various courses here, which are on bioinformatics, multi-omics, structural biology. Multi-omics includes genomics, epigenomics, transcriptomics, and metagenomics. And then we have a course for structural biology that you are supposed to go through as an online associated resource, and also a course book on data science, which uses R and Python for biomedical data analysis. So by clicking on the course tag, you will be able to view all the courses up here, and then you can pick the one that you would like to go through and learn more about it. Same goes for the example project. Simply click on the project tag and the list of all the example projects will be listed. Now, these example projects are actually applications of the coursework that you will be going through, just to have an idea that how the concepts, methodologies, and guidelines to run various pipelines on the server could be applied to answer a particular research question in hand. So that was it about the example projects and coursework that you can go through. And now talking about the structural biology and cheminformatics coursework that you're supposed to go through. So as this program begins, we have uploaded the first lesson for the program. And this particular lesson is a free lesson that you can definitely go through. And as we'll be proceeding along with each and every session of Cheminformatics program, you'll be able to see the other lessons here as well, which you can go through, practice, do the hands-on practical, and take your questions. Now, talking about the Slack channel, so this is the Slack channel that I was talking about. And the program participants will be a part of our Slack channel. Now, this one is a general group, but when we have a particular program going on, we have a group like this where all the program participants are here and also the mentors, and you can clarify questions here in the group itself. Okay, so those who are asking for a fee waiver or scholarships, you can email Sparshadhar at marketing@gmail.com. Sorry, omicslogic.com. And she'll be able to help you with this. Also, I'll quickly put the scholarship form in the chat box. Now, when you will mail Sparshadhar at marketing@omicslogic.com, please let her know which subscription level are you interested to pick. And yes, thank you, Sparsh, for sharing the contact number. So we have an Indian contact number where you can call and have a word with Sparsh, and she'll be able to help you out with your enrollment. So that was it about registration and also about the portal and resources that you'll get the access to. Now, if there are any other questions, you can please put them in the chat box and I'll be happy to take them up. Yes, till then, I'll just quickly share the link to the form. Manpreet, for now, this is a one-time course where we are having an introduction to Cheminformatics. And next month, we are going to have an advanced level of course where we'll be taking up more advanced topics and also dig more deep into the hands-on practice. Right, so this is a coursework where we are getting started with Cheminformatics, and also we'll be discussing various aspects. And those who are just about to begin with this particular field, I think this is very nice for them, and also for those students or researchers who want to understand more of the concepts and listen to these industrial experts. I think this is the best program for them. And after this, we are going to have an advanced level of coursework. So what I think is that going through this particular coursework will lay off a very nice foundation for all of you for the interested participants to take up the advanced course, right? And for now, we have no plans of running this maybe later, but we'll definitely update you once we have such a plan. Okay, so by talking about the certification process, when you talk about how well certified these courses are globally, so we are not definitely going to rate it up, but yes, I think that these certifications are globally accepted as they are certified by the most industrial experts and people who I think have the best knowledge out of it. And to highlight more about the certification process and its global recognition, I think Dr. Raghav can answer it much better than me.
Manpreet, yes, I mean, we, as it has already been clarified and as Sonalika covered, that we are involved with variety of different universities all around the world, and this certificate is globally valid. But what this certificate will give you in terms of skill, in terms of learning experience is, you will learn the necessary skills, not just and get a certificate for the validity of those skills. If you include the skills that you have learned and the courses that you have learned in your curriculum or in your resume, then you will have a valid certificate to show for, and also knowledge that you have acquired in the process of going through this program and in the process of going through these courses will speak for itself. And we value that even more, or even at a higher rate than you gaining certificate. By a certificate from us will ensure that you have learned all the necessary skills, and you will be able to converse in the language, or you will be able to understand all the necessary methods in any cheminformatics document or a paper, research paper, so that when you are going to apply for new jobs, and you'll be able to, for example, think independently in these using these algorithms and using these processes. That is what we will prepare you for, and that is what this program as an introduction will help you to gain more insights. And the advanced program that Sonalika is going to talk about is going to give you a lot more practical experience and a lot more understanding of which methods to choose for which question and which data. So the methods, the question, the data, all of them, you will have a lot of experience and a lot of understanding in the choices, and also in designing a project or the analysis methods. So I think that will partially answer your question. And yes, many people have been using these certificates, and they are valid, and they are highly regarded in the job market globally. So any other queries regarding to that?
So Manpreet, as I explained, that we have 3 different levels. It's not that you definitely have to pick the advanced level or $180. You can pick the one that works out for you. When you talk about the access of resources, that is session videos, like the recordings, the associated resources, all of them are going to get the access to it, definitely. But the access is different just in terms of mentor availability, right? So in the beginner level, you can definitely pick up your questions in the Slack groups. But yes, to have one-on-one meet with the mentor, intermediate would be the best, I think. And advanced is for those people who actually want to join, let's say, postdoc, they want to join a postdoc position or a PhD program. There are many students as well who want to join some master's program, and they are supposed to submit a research proposal or maybe start with a research project. And like work on it, and before that they need to submit the proposal to us as well, right? So going through this literature review and coming up with a nice good proposal to work on your research project and get started with it is what I think. People having this sort of goal, they should pick the advanced level, right? And also throughout these sessions, it's not just the theoretical knowledge that you're going to get, you'll also go to the practical hands-on analysis. But if your ultimate goal is to get a research project out of this particular training program, then I think you can pick the advanced level. And still, if you want to pick the advanced level, but you think the cost is way too high, I have sent you the email ID in the chat box. Please contact Sparsh Thar, and she'll help you out with getting some good, nice scholarship for the advanced level. Yes. And perfect. Any other questions? All right, if we have no more questions, I think we are good to end today's session. And we'll be meeting tomorrow now, and we'll hear from Dr. Puneet. And he'll be talking about cheminformatics. We'll get you all started with introduction to cheminformatics first of all. And we'll also talk about various tools and databases tomorrow, and go through some of the tutorials, and also learn more about the file formats. So let's now catch up tomorrow, the same time. Have a nice day. Thank you for joining.